Welcome back. In this lecture, we are going to start our new module on Pangenome graphs. And specifically in this lecture, I want to introduce the topic of pangenomes. So let's get started. So far we have covered 7 modules, and in this module we are looking at the module 8, that is Pangenome graphs. So what is a pangenome? Well, the broad definition of pangenome is following. So when we say the word pangenome, we usually mean a collection of genome sequences from multiple individuals within a species or a clade. So broadly we are looking at a collection of genome sequences, and this happens to be also one of the active research topics. So for example, if we look at the number of research articles that mention this keyword pangenome, then you can see that this topic has emerged as more popular topic very recently. So the number of publications have increased as we have gathered these statistics from the PubMed database, and this is partly because of the availability of high quality genome sequences has more recently increased for both human as well as non-human species. Now, given the large collection of genome sequences available for many species, it is useful to analyze these sequences collectively through a pangenome based analysis. Now, specifically for humans, the motivation for this pangenome analysis comes from the fact that if we just look at the genome sequences of 1 or 2 individuals at a time, then that gives us an incomplete view of the genetic diversity. For example, if let us say we focus on a specific region of the chromosome 6 of human genomes. So this is the MHC, which is the major histocompatibility complex region of the chromosome 6, and this is approximately 5 million characters in length. Now if we look at these sequences of multiple individuals and see how similar they are, then what we usually find is that these regions vary significantly from one individual to other, and therefore collectively looking at the sequences and analyzing them sequences tells us how this sequence is evolving in different individuals and what might be the relationship of different sequences with different phenotypes. So for specifically for the MHC sequences, here is a visualization of how these sequences align to each other when we consider 8 sequences from 8 individuals. So this is a visualization of the alignment of 8 MHC sequences from different individuals. In different colors, you see the homologous or similar segments across the sequences. So for example, you can see that the left hand side of the sequences shown in red is well conserved across all 8 individuals. Similarly, the rightmost side is well conserved in all individuals. However, if you look at the middle portions, this is where you see a lot of changes happening from one individual to other. So these colored segments indicate the similar segments across different individuals, and these lines indicate the relationship across different individuals. So this is a common way of analyzing multiple sequence alignment, and this is a motivating example to show you that some regions of the human genome are highly polymorphic in nature. That is, they tend to vary a lot, and if we just look at one sequence or one or two sequences, then that would give us an incomplete picture of the genetic diversity present in human populations. So let us look at the timeline of how this pangenome topic has emerged over the few years. Especially, so this timeline is in the context of human pangenomics, where we know that in 2001 was when the first draft of human genome was published. 2003 happens to be when the first mention of pangenome term appeared in the literature, and the first mention of human pangenome appeared in 2010. However, as you can imagine, to do a proper pangenome analysis, we also need high quality genome sequences of individuals, and for a long time it was not possible to get high quality genome reconstruction of human genomes because of the limitations in sequencing technologies. Very recently, the improvements in sequencing technology, for example, the ability to sequence long and accurate reads, dramatically improved the quality of genome reconstructions, and following that was also when the first complete and gapless assembly of a human genome was published. Now, because of these improvements in assembly quality, and because now we have access to high quality human genomes, in 2023 was when the first draft of a human pangenome was published. This draft human pangenome comprised genomes of 47 individuals, and these are high quality genome sequences assembled using latest sequencing technologies. So, as I said, the concept of human pangenome was catalyzed by the ability to sequence high quality human genomes. Now, we can also look at another statistic to see how the quality of assemblies has improved over the last few years. So, for example, on the y axis you see the average length of assembled genome sequences indicated by their average length in million base pairs, million characters. So, this is a log scale, and if you see over the years as different technologies emerged in the market. So, for example, Sanger, Illumina, PacBio, Oxford Nanopore and so on. As sequencing technologies have progressively improved, the quality of genome reconstruction has also improved accordingly. So, more recently the contiguity of genome sequences assembled de novo is significantly better than the quality of sequences that was possible 10 years ago. So, given this improvements in genome assembly, where we are able to assemble genomes that are either complete or near complete, and when we say complete we mean end to end reconstruction of each chromosome of the human genome, then that is when the analysis of pangenome makes sense, because that is when we have access to high quality genome sequences. So, now looking at the various ways of representing these pangenomes, we will find that in the literature people have been using different ways of representing these pangenomes based on the application they are addressing. So, one of the simplest ways of representing a pangenome is basically simply using a Venn diagram, where we can say we can analyze the genome sequences of multiple individuals. Especially this is more popularly done in the context of microbial genomes, where different genomes tend to have different set of genes. So, if we look at the individual genomes coming from a single species, then the core genome is the portion of the genome sequences that is universally present in all the sequences. So, in this case the core would be the central part, which is the core part which is present in all the genome sequences. So, this central part of the Venn diagram is telling you the sequence composition that is present, universally present in all the individuals. The other component is the accessory genome, which is only found in a subset of individuals. So, this analysis is useful because in research we want to analyze the core genome as well, where we may want to see the set of genes that are present in all individuals, which represent the genetic information needed for basic survival. Whereas the accessory genome would usually correspond to the set of genes that would allow the subset of those individuals to adapt better to specific environments or conditions. So, in other words, the core genes would be housekeeping genes which are needed for survival, and the accessory genes would give certain advantage to specific individuals. So, this is the simplest analysis that one can do with pangenome, and this is more popularly used for microbial, bacterial and viral genomes, where we want to understand the set of genes differently based on whether they are core or accessory. Now, a more detailed or fine grained representation of pangenomes is done using graph based models. So, one simple model would be using a De Bruijn graph based approach, where remember that from our earlier lectures where we defined the De Bruijn graphs, where the De Bruijn graphs comprise vertices or nodes which correspond to the k-length substrings observed in the sequences. So, these are the k-mers observed in the sequences, and in this case the edges would correspond to the k minus 1 length overlaps among these nodes. Now, for example, let us say that we have 3 genome sequences coming from 3 individuals. So, let us say this is the first sequence, this is the second sequence, and this is the third sequence. Now, a graph model to represent a pangenome would be to construct a De Bruijn graph or a colored De Bruijn graph in which all the k-mers observed in the sequences would be nodes, and we would connect them if they overlap using k minus 1 characters. So, at the bottom I am showing you a toy De Bruijn graph constructed using tumors. So, if you consider all the tumors available in these sequences, that is AT, TG, GC, CG and so on, and construct a De Bruijn graph, then we would get the following De Bruijn graph. And in this graph we can also color the individual edges. For example, if I see that the overlap between AT and TG vertices was observed in genome sequence 1 as well as genome sequence 2 as well as genome sequence 3. So, in this case I would color the edge with yellow, red and blue colors. In some cases, for example, this overlap AGC is observed only in the genome sequence of second individual. Therefore, observe that this edge is only colored with a red color. So, this De Bruijn graph with additional color information on the edges is referred to as colored De Bruijn graph because of the coloring done on edges. We could have also done coloring on vertices, and that is just a matter of choice of how the model is defined. Now, so this is one way of representing these pangenomes using a graph based model. There is also another graph based model which is commonly referred to as pangenome graph, and it is more prevalent in recent literature. So, in this pangenome graph or a sequence graph, we basically have a graph model in which we would have a directed graph where nodes are labeled with DNA sequences and edges imply adjacency of those nodes in one or more genomes. For example, at the bottom I am showing you this example of a pangenome graph constructed using genome sequences of 4 individuals. So, here you can see that this graph has 13 nodes. The first node is labeled with sequence ATC, the second node is labeled with sequence C, the third node is labeled with sequence T and so on. And we would connect the 2 vertices when we find that these 2 sequences are adjacent in the underlying sequence. and observe that each of these individual sequences would be spelled as a path in this graph. So, for example, I can the genome sequence of individual 1 can be spelled as a path in this graph. So, this goes from here to here to here to here to here and to here, right, following this dark blue color. Now, it is observed that in this graph basically what you are observing is that the changes or the differences in the genome sequences are appearing in the form of bubble like structures, and wherever you have conserved sequences that are universally present in all the genomes, for example this node as well as the final node, that would be collapsed into a single vertex. So, the sequences shared among the individuals are represented using a common vertex, whereas the sequences that are varying in these genome sequences are presented as alternative paths in this graph. So, this is in some way a compact representation of all the genome sequences, and at this point we can ask, okay, how, given the 4 genome sequences, how did we build this graph, right. So, to get this intuition, again I am using these examples to give you intuition. Maybe in the next lecture onwards we will start looking at some theory. So, let us say you have 3 genome sequences, and as a first step we would want to know from these 3 genome sequences what are the sequences that are conserved across all the individuals and what are the sequences where we see mutations in these genome sequences. Now, one way to analyze multiple genome sequences to understand which portions are similar and which portions are variable in these genome sequences, one way is to do multiple sequence alignment. So, in our lectures we have seen pairwise sequence alignment, and this is multiple sequence alignment is a generalization of that problem where you are aligning multiple sequences together at a time. Now, the multiple sequence alignment would tell us that this portion of the sequence is conserved across all individuals, whereas there is a portion of the DNA sequence which is varying among the individuals. So, for example, here you see mismatches as well as insertions and deletions, right. So, columns where equal characters are present in all the rows, that is where we would say that it is well conserved, whereas the regions where we are observing several insertions, deletions or mismatches, those would correspond to the variable regions in the genome. So, now once we have this multiple sequence alignment representing the regions of homology and regions of differences in these genome sequences, what we can do is we can use that multiple sequence alignment to construct a compact graph. For example, if we know that this portion is well conserved, then we would use only a single vertex in the graph to draw the pangenome representation. Now, for the variable portion where the sequences are differing a lot, we could use different vertices for individual sequences to indicate that these sequences are now varying from each other, and therefore, they should be represented as alternative paths in the graph. Similarly, again observe that we have collapsed 2 sequences into one vertex, because here we see that the last 3 characters are conserved among first 2 individuals. Therefore, again we would use a common vertex to represent that. So, the overall motivation is to represent the individual genome sequences compactly, so that the conserved sequences are represented using a common vertex and the variable regions are represented using alternate paths in the graph. Okay, so let us move forward, and so this is a better visualization of these graphs, and basically what we are saying is that whenever we see these alternate paths, that is, for example, here you see an alternate path having 2 different routes, one route having label T and another route having label A. So, in this case these alternate paths would often appear only when there is mutations happening in the genome sequences. So, this gives us a collective view of how these genome sequences are related to each other as well as how they are different from each other. Right. For example, here is another view where one of the individuals have a insertion of a sequence, and there you can see that alternate path coming up at the bottom. Now, what are the applications of pangenome graph? Like, why would we want to use these pangenome graphs for human genome analysis? Well, one of the key applications of these pangenome graphs is genome resequencing. For example, when we remember that when we sequence a genome sample, we would normally get millions of these fragments of the genome that we call as reads, and these would be the fragments of the genome that have to be further analyzed to understand the target genome sequence. Now, one way to analyze the genome sequence is to say that okay, we will take all these fragments and we would do the pattern matching of these fragments to a reference template. So, we would take a reference template and we would pattern match all these fragments on that reference template. So, this would be referred to as read mapping, where we are mapping all these reads to the reference genome. Now, the purpose of this mapping would be to say okay, if whenever these reads are getting mapped and we see some difference in between the reads and the reference genome, there is where we know that the mutations are happening in the genome sequence. So, from the differences in the read alignments, we would know that there is mutation, the underlying the target genome had mutations at 2 positions. So, we would call these mutations and we would say that at position 11 there is a mutation from T to A, and similarly at position 23 there is a mutation from G to T based on these read alignments. Now, one limitation of this approach is that when we are doing pattern matching of these reads to the reference, we assume that these reads are approximately similar to the reference genome. That is, these reads would align well to the reference at some location, and only the small differences would be called as mutations. However, what if there are polymorphic regions where there is a significant difference between the reference and target genome? There what will happen is that we may not be able to map reads at all, because there is no good similarity between the reads as well as the reference, and it may be difficult to do matching of those reads to the reference at those locations. So, for example, I was giving you this example of the MHC region in chromosome 6 in one of the earlier slides. Those are the regions where we may see poor mappability of reads. For example, if the read there is a significant difference between the target genome and the reference genome, then we may not be able to match reads in those polymorphic regions. So, this is where the pangenome references are promising, because what we can do is we can replace this reference genome, and instead of having a linear representation of the reference, we can have a more richer representation where the reference comprises genome sequences of multiple individuals. So, that for every target genome we have some region of similarity in the pangenome graph. Now, this is also makes sense because ideally a reference genome should represent the complete diversity of human populations. So, in some sense, if the reference genome is primarily derived from, let us say, a single individual, then we are biasing the genome analysis of all the target samples. So, this is an active research topic. Also, for example, if you want to see a real pangenome graph, so far we were looking at toy examples, but this is how a real pangenome graph looks like in the MHC region. So, the region that we were looking at in the previous examples, which was highly polymorphic between different individuals, this is how the graph of that region looks like in practice. So, this graph is constructed using genomes of 47 individuals from this publication, and you can see that if you look at just the linear reference of the human genome, then this linear reference is just a single path in this graph represented in the green color. There are other genome sequences which are significantly different from the linear reference, and those are appearing as different paths in this graph, right. So, by representing this genetic diversity, the expectation is that this pangenome graph would serve as a better reference, because now when we are resequencing a genome, we would have different sequences available to us where pattern matching can be done. So, then the idea would be that okay, when we do genome resequencing, we would do read mapping to a pangenome graph, and we would use that pangenome graph for calling variants in the target genome. So, this was recently demonstrated that the use of pangenome graph also leads to better precision and recall for variant calling. So, in this plot I am showing you the precision recall curves of variant calling accuracy obtained using 6 different methods. And in this plot, basically as you can imagine, the more top, basically the topmost and right and top corner is the ideal place where you want high recall and high precision at the same time. So, in this plot, basically the top 4 approaches are all based on using a pangenome graph. So, this HPRC is using the human pangenome graph that we were just looking at, and this dragon graph is a graph used by a proprietary software called Dragon. And in all 4 cases, we can see that compared to the approaches that rely on a linear reference genome, the approaches that use a graph based pangenome reference achieve a higher F1 score. And this is mainly coming from the polymorphic regions in the human genome where the read mapping using linear references can be inadequate. So, as it turns out, this is also a very recent and active research topic. So, for example, problems like constructing pangenome graphs for human genomes or mapping reads accurately to these pangenome graphs, as well as calling variants in using these pangenome graphs, all of these are active research topics with publications appearing frequently in the literature. So, we will close this lecture here. So far we saw an introduction of the pangenome topic. I showed you various motivating examples to illustrate why pangenome graphs are being used recently in the literature. In the next lecture, we will look at 2 topics related to pangenomes. One is multiple sequence alignment, where we want to align genome sequences coming from multiple individuals together, so that we can understand the regions of conservation as well as regions of differences accurately. The second topic in the subsequent lecture we will look at is sequence to graph alignment, which would be useful for mapping reads to pangenome graphs. Thank you.